I'm Mark Pegram. I'm the Associate Director for Clinical Research at the Stanford Comprehensive Cancer Institute and a member of the Division of Medical Oncology. PARP is simply an acronym that stands for poly-ADP ribose polymerase. Poly-ADP ribose polymerase. So you can see why we call it PARP instead. It's a mouthful. It's simply an enzyme that's involved in DNA repair. The main role of PARP is to detect and initiate immediate cellular response to single-stranded breaks in the DNA. Recall that DNA is double-stranded, i.e. the double helix, and sometimes there can be damage to the DNA resulting in nicks in one of the strands. So once PARP detects a single-strand break like this, it binds to the broken DNA and then begins the synthesis of a polymer called poly-ADP ribose. And this polymer is like a group of sugars, ribose sugars, that act as a flag that then recruits other DNA repair machinery to the broken DNA. And in concert, this whole DNA repair complex, flagged by the PARP poly-ADP ribose, acts to repair the damaged DNA. Now, let me digress just a little bit and talk about another type of DNA repair that repairs double-stranded DNA breaks. BRCA1 and BRCA2 are proteins that are important to repair double-stranded DNA breaks by a process called homologous recombination. When genes for one of these proteins is mutated, the function of the BRCA protein is knocked out, which can lead to errors in DNA repair that eventually cause cancer. Breast cancer, as well as other cancers, such as ovarian cancer, prostate, pancreatic, and others. Alan Ashworth, a British-born molecular biologist, and now Cancer Center Director at UCSF here in San Francisco, was a key part of the team that in 1995 discovered the BRCA2 gene. So Ashworth became interested in how he might selectively kill tumors that harbor these BRCA mutations that can be inherited through families, leading to these familial cancer syndromes that you are well aware of. So he borrowed a phenomenon from the genetics field called synthetic lethality. Now, what synthetic lethality means is when a combination of defects in two different genes leads to the death of a cell, whereas deficiency in only one or the other does not. This was first discovered in fruit fly genetics by Calvin Bridges back in 1922. An easy way to understand the concept of synthetic lethality is the analogy of the three-legged chair. The idea is that a cancer cell with a broken leg, if you will, like a broken BRCA gene, is hobbled, akin to the three legs. Remove another key gene from the chair, and the chair will topple, and the cancer cell will die when it gets down to two legs. So Ashworth constructed a high-throughput synthetic lethal screen in the laboratory where he took BRCA mutant cells and then systematically knocked out all of the other genes one at a time in the genome, and found that when the gene for PARP was knocked out, BRCA deficient tumor cells could not survive. So the mechanism behind this synthetic lethality in the case of PARP and BRCA genes is that when PARP is knocked out, I told you that it's responsible for repairing single-stranded DNA nicks. But it turns out that if these nicks persist unrepaired, for example, if you would block PARP with a drug, then when the DNA is replicated in that cell, the single-strand breaks are turned into double-strand breaks, which could ordinarily be fixed by BRCA proteins because they can fix double-strand breaks. But if BRCA is mutated and BRCA function is broken, then it can't repair the PARP-induced DNA damage. So PARP inhibitors work uniquely in BRCA mutant tumors. Normal cells that don't replicate as quickly as tumor cells by and large, their DNA doesn't convert the single-strand nicks into double-strand nicks following PARP inhibition, so they are able to tolerate PARP inhibition better, although blood cells are affected by PARP inhibitors because PARP inhibitors can cause anemia, low white blood cell counts, etc., as a known side effect. Olaparib and talazoparib are examples of PARP inhibitor drugs, which received FDA approval for the treatment of patients with inherited BRCA mutated metastatic breast cancer. Data from randomized clinical trials in patients with BRCA mutations show that these drugs are even better than chemotherapy as first-line treatment for metastatic breast cancer. This is a triumph of modern translational research and drug development based entirely upon first principles established by the field of genetics, a tour de force of bench-to-bedside discovery research. Meanwhile, more recently, there is some excitement around the use of PARP inhibitors, not only because of their activity against BRCA mutations, but also because they appear to modulate immune responses. In particular, there's a lot of interest in combining PARP inhibitors with immunotherapy agents. There are several ongoing studies to assess whether this phenomenon may enhance activity of immune checkpoint inhibitors. And if this approach is successful, the role of these agents may be expanded beyond that of just BRCA mutation carriers in the future.